We have seen the benefits of bacterial overexpression systems. However, using bacterial systems to express human genes comes with certain complications. Let's look at some of these obstacles and see how they have been overcome. A major challenge to expressing eukaryotic genes in a prokaryotic system is the presence of introns that must be spliced out of the original mRNA molecule. Secondly, mammalian proteins are also subject to post-translational modifications that would not be possible in a bacterial host. Let's look at the example of the insulin gene to see how it was successfully overproduced in a bacterial host. As you know, the nascent mRNA transcript is first processed by the spliceosome to give the mature mRNA transcript. You may recall that insulin is a protein that is subject to significant trimming after translation in order to give a fully functional form. The original form, pre-proinsulin, is first trimmed to give proinsulin. This is then cleaved to give separate A and B chains that are joined by disulfide bonds. None of these processes could be accomplished by a bacterial host. The first step in the cloning process was to synthesize DNA in the lab to give separate A and B chains. These were then inserted into bacterial plasmids, as shown below. Separate strains of E. coli were then transformed with one of the two plasmids, as illustrated here. The protein products were isolated from the bacterial hosts and then combined to form the functional human hormone, insulin. We've seen that PBR322 and PUC19 can be used to insert foreign DNA and amplify it. However, if we want the DNA to be transcribed and translated, an expression vector is needed. These are plasmids that have components to allow the processes of transcription and translation to be completed in the bacterial host. This means initiation and termination sequences for both processes. Of course, we need the cloning attributes already described, an origin of replication for the plasmid, a multiple cloning site where we can insert the gene, and at least one selectable marker. In addition, the gene must carry a promoter that can be recognized by the host RNA polymerase so that the gene can be transcribed, and a termination recognition site. PET11 is one such expression vector. These techniques have allowed scientists to prepare many proteins in bacterial systems, allowing for higher production and lower costs of many hormones.